Okay, hello everyone. What I'd like to do now is talk a little bit about cells, cellular organization, and organization of the genetic material within those cells. And this is really, again, just to provide a little bit of background, a little bit of terminology that will allow us to discuss the processes of mitosis and meiosis, two mechanisms that cells use when they're dividing to segregate replicated genetic information into the daughter cells. So the first, I guess we'll start from the beginning here, is the idea that life as we know it on this planet is cellular based. And there are two fundamental types of cells that you find in living organisms. There's what we call a prokaryotic cell and a eukaryotic cell. So these cells share common features, very basic features. And those features are that they are on the outside surrounded by a plasma membrane. And that plasma membrane then holds a gelatinous material that we call the cytoplasm. And within that cytoplasm, there is genetic material, and predominantly in the form of double-stranded DNA, either a single molecule or multiple molecules. And it's that genetic material, again, that carries all of the information that allows for the maintenance and metabolic activities to occur and cells to divide. It allows for the cell to exist as a living unit. So in addition to that, a common feature of these two cell types are what we call ribosomes. So again, ribosomes are a very important piece of cellular machinery. Those are structures within the cell that allow you to utilize genetic information. So some of that genetic information allows for or encodes information for protein production. And the proteins are actually manufactured on these ribosomes. So these are the common features between prokaryotic and eukaryotic cells. There are, fundamental biochemical differences and so on, but we're not going to look at it that deeply. So what is the basic distinguishing characteristic between prokaryotic and eukaryotic? Well, it's simply the idea that in a eukaryotic cell is that the genetic material is compartmentalized. So it's housed in a second membrane-bound compartment, not the outer plasma membrane, but instead a nuclear membrane. And it's within this subcellular compartment that the genetic material is found, again, in the form of either one or more DNA molecules. So that's true for eukaryotic cells. That's not true for prokaryotic cells. Prokaryotic cells, what you find is that the genetic material is out in the cytoplasm, but it's within a specialized region of the cytoplasm that we call a nucleoid. So again, it's not your typical cytoplasm, I guess, but there's no surrounding membrane that encloses or compartmentalizes that genetic material within the prokaryotic cell. So here's a representation of the six kingdoms of life as we know it. Again, all cellular based. So down here we have the true bacteria. Eubacteria are the archaea. Again, these are the two prokaryotic forms of life that are comprised of single cells, unicellular life forms that have that basic prokaryotic type organization. Again, there are distinctions, again, biochemical and otherwise, and even at a genetic level, that distinguish the true bacteria from the archaea. But again, we're not going to go into any particular details about that. So then the rest of the kingdoms of life, the other four, are eukaryotic life forms. So again, comprised of cells that have the eukaryotic type of organization. So the protists are sort of the oddballs of nature. They're eukaryotic. The protists can be unicellular or multicellular. They can exist in both forms. There are some instances where you can find more than one nuclear compartment that carries genetic information. So like I said, sort of odd life forms on the planet. Then you have the remaining three eukaryotic kingdoms, the plants, the animals, and the fungi. All of these are multicellular life forms, again, no longer unicellular. And all of them consist of this eukaryotic cell organization. So here's a generic representation of a bacterial cell, representative of a prokaryotic system, a prokaryotic cell. So here's the outer plasma membrane. Here's the cytoplasm that's enclosed by that membrane. And then lastly, you have a chromosome. And again, in prokaryotic organisms, predominantly what you see is that there's a single molecule of DNA that represents the genetic material of the organism. And what you find is that genetic material, again, is in the form of a DNA molecule, and that DNA molecule is circular. So here's, again, a representation of a prokaryotic genome visualized under an electron microscope. So again, you can see the circular nature. You can see that it's essentially naked DNA. There are proteins that are associated with that DNA molecule, that chromosome. However, it's not extensive. Okay. So again, this would be true. This type of organization for the genetic material in a prokaryotic system is true for the true bacteria, the eubacteria, as well as the archaea. The only slight variation is sometimes certain types of bacteria or prokaryotic systems, you will find more than one chromosome, but it's very rare. And again, they're always localized within that nucleoid region of the cytoplasm. So that organization is in stark contrast to here. What we have being represented is a generic eukaryotic cell, and more specifically an animal cell. So here's the outer plasma membrane that encloses a cytoplasm, and what you find is that within that cytoplasm there's many different structures, many different organelles, all of which carry out unique and distinct functions. Okay, so this is in addition to the ribosomes. So we describe that as being, the one type of cellular machinery that you find in prokaryotic systems. Well, they're certainly found in eukaryotic cytoplasm as well. However, you find many other organelles, many other structures as well. So again, not to go into any particular detail, but here's a Golgi apparatus, endoplasmic reticulum, a tubular network that runs throughout the cytoplasm. There's these centrioles down here. There's a paired set of centrioles that are found within a region that's called a centrosome. And again, that's an important component of the machinery that allows for cells to segregate genetic information through a cell division. So we'll talk more about them in a little bit. In addition to that, here's a lysosome. And then lastly, mitochondria, the powerhouse, the energy producer within the cell. I should point out is that the ribosomes in the eukaryotic system, you can find some free ribosomes, but they're predominantly associated with the endoplasmic reticulum. So then finally, well, where's the genetic material in this eukaryotic system? So as I said, it's in a membrane-bound compartment, a nuclear membrane, and all of the genetic material is found within the nucleus. Okay, that's not completely true, and we'll talk more about this as we go along, but the bulk of the genetic material that you find in a eukaryotic system is within the nucleus of the cell, and that's really the type of genetic information we're going to focus on. In particular, we're talking about mitosis and meiosis, and when we talk about Mendelian genetics. But I do want to point out at this stage here is that mitochondria carry their own unique and distinct genetic information as well. And actually, that genetic information is organized much more closely with what you see in a prokaryotic system. again, that, as I pointed out earlier, a circular molecule of double-stranded DNA. So here's another generic eukaryotic cell. This is a plant example. So it's basically the same as what I just pointed out for the animal system. There's an outer plasma membrane. Plant cells are generally, once they're produced through cell divisions, they're cemented into place with a cell wall. You don't see that in animal systems, but in any event, there's a plasma membrane that encloses a cytoplasm. In that cytoplasm, you find, for this plant cell, all of the same types of organelles, cellular machinery that I discussed or described for the animal cell, but there is a unique organelle in plants. This is what we call a chloroplast, and again, a chloroplast is a site of photosynthesis, allows for CO2 fixation. So again, an organelle, cellular machinery that is unique to plant systems. And again, the reason I just mention it at this point is the idea that chloroplasts, like mitochondria, carry their own unique and distinct genetic information. It seems to be organized more in a prokaryotic form as opposed to a eukaryotic form. So in any event, just like the animal cell, here's the nucleus of the plant cell, a nuclear membrane, and it's within that subcellular compartment, the nucleus, that you find the genetic information, or the bulk of the genetic information within this plant cell. So just to give you an idea of the greater level of complexity for these eukaryotic chromosomes compared to what I described for the prokaryotic chromosome earlier, these two representations here are human chromosomes as viewed under the electron microscope, different ways of preparing the chromosomes so that they can be visualized under an electron microscope. So just looking at them, so first of all, what do we notice that's different about them? Well, they're linear as opposed to circular, as I described for the prokaryotic chromosome. And they're also much more complex in their organization. So in other words, it's not just a DNA molecule with a little bit of protein associated with it, but a lot of protein. It gives it this very complex organization here. And so we'll talk a little bit more about that as we go along. I just want to point out the greater degree of complexity in terms of how the actual genetic material is presented within the nucleus of a eukaryotic cell as opposed to the nucleoid of the prokaryotic cell. So again, the way that the DNA is organized is fundamentally different in the two systems. So down here, actually, what we have is, again, these are representations of eukaryotic chromosomes. These happen to represent, these are individual human chromosomes up here. This is actually a collection of 8 chromosomes that are represented within the cells, the nucleus of the cells of a fruit fly. And so again, they've been prepared in such a way that they can be visualized. And the point here is simply that they have a complex structural organization to them. They're not simply DNA molecules. You can see they're linear again, but they're not DNA molecules that you have very little protein. There's lots of proteins that are associated with those chromosomes. So a much more complex organization than what you find in prokaryotic systems. So what do we know about these chromosomes, whether we're describing a prokaryotic or a eukaryotic? I've been describing it as they actually serve as the housing in the cell for, the chromosome is the housing for the genetic material itself in the form of a DNA molecule. Well, where did this idea that genetic material is associated with chromosomes, specifically within the cell, where did this idea come about? Well, it actually came about through work of these two individuals independently, Walter Sutton and Theodor Boveri. They both proposed in the early 1900s this idea of chromosomal theory of inheritance. In other words, drawing the correlation between Mendel's work, Gregor Mendel's work, that described inheritance patterns for traits in the pea plant with chromosomes. So in other words, being able to explain Mendel's observations based on segregation patterns that occur for chromosomes in the eukaryotic system that is the pea plant. Okay, so they were the very first. We certainly know that as a fact nowadays, and we've known it since the early 1900s. But these were the two individuals that first drew this correlation. So specifically, the genetic material, Mendel's very abstractly defined genes being localized specifically within the cell, within the chromosomes, okay, in the way that I've described for both prokaryotic and eukaryotic systems. So again, just as far as a definition, a chromosome is a structure within the cell that carries this genetic information, and specifically in the form of a linear DNA molecule for eukaryotic systems, a circular DNA molecule in the case of prokaryotic organisms. And there's a fundamental difference in terms of levels of complexity in terms of the organization of those chromosomes when you look at a prokaryotic chromosome versus a eukaryotic chromosome in terms of, associated materials, the proteins and other things that would be associated with the DNA molecule within a eukaryotic chromosome. So again, this is just a slide that summarizes all of the previous points that I've made, so you can read through that on your own. But what I do want to point out here is in eukaryotic systems, when you look at the number of chromosomes that you observe within the nucleus of the cell of different types of eukaryotic organisms, there's a tremendous amount of variability. Okay, so that's very different from the prokaryotic system, where I said predominantly what you find is that there's a single molecule of DNA that represents a single chromosome within that prokaryotic cell. There are exceptions to that, but that's the predominant observation. But in eukaryotic organisms, when you look at the chromosomes within the nucleus of the cell, there's a tremendous amount of variation when you look at different organisms. And the point that I want to make here is that there's no correlation between complexity of the organism and the number of chromosomes that you find within the nucleus of the cell. So here's a table. This is summarizes, again, chromosome counts from cells of many different types of eukaryotic organisms. And what I want to point out here is that actually these chromosome numbers, this is what is referred to as a haploid number. So in a eukaryotic system, and we'll get back and talk about this a little bit more detail in a few minutes. But when you look in a eukaryotic cell, typically you find 2 copies of every chromosome that is unique within that nucleus of the eukaryotic cell. So that's what we call a diploid state. So a haploid state you'd have 1 copy of each unique chromosome that you would find within the nucleus of the eukaryotic cell. So as for definitions, diploidy, sometimes referred to as a 2N state. You have 2 copies of every chromosome in the nucleus of the cell of the organism, and therefore 2 copies of every gene. And then the haploid state would be 1 copy of every unique chromosome that's representative of that particular organism, and therefore only 1 copy of every gene. So here we're looking at a haploid number. So for corn, it's 10, chimpanzee, it's 24, humans, 23 unique chromosomes. But the 2 that I really want to point out here are the mosquito and the waterfly. So again, in terms of body plan, in terms of structural organization for these 2 different insects, these 2 different invertebrates, the mosquito and the waterfly, I think everybody would agree that they're very similar in terms of the level of complexity of that body plan. However, if you look at the haploid number of chromosomes in the cells of the mosquito compared to the waterfly, there's a tremendous difference. So the mosquito, the haploid number is 3, and for the water fly, the haploid number is 80. So again, the idea that there doesn't seem to be a correlation between chromosome number for the eukaryotic organism and the complexity of that organism. Okay, so just another bit of terminology here, this term genome that's been thrown around, was first coined, I think it was in the late 1980s, early 1990s. The genome is a term that describes the collection of chromosomes within the nucleus of a eukaryotic cell, or even the single chromosome of a prokaryotic cell. So it's simply the complete collection of all of the chromosomes of the organism, consequently the complete complement of all of the genetic material and genes carried by that individual. So you can describe the genome either as a haploid genome, again having one copy of every unique chromosome within that cell, or you can describe it as a diploid genome. You see it used both ways, but in both cases, you're describing the complete collection of all of the chromosomes and consequently the complete complement of all of the genes that are found within the genome of that particular organism. Okay, so here's an example of a human genome, and it's organized in a format that we describe as being a karyotype. So again, if you remember from that previous table that I just showed you, the haploid number of chromosomes in the human genome is 23. So a diploid genome would consist of 46 chromosomes. So within this karyotype, if you count up all of these individual chromosomes, you'd count a total of 46. And you can see that these chromosomes exist as pairs within the genome. Okay, so in other words, the 46 chromosomes actually are represented in the form of 23 pairs of unique chromosomes within the human genome. So if you look at chromosome pairs 1 through 22, and you were looking at a karyotype, they would be indistinguishable whether you're looking at the karyotype of a male or a female human. Okay, so these are what we call autosomes. Okay, again, autosomes are in terms of being able to suggest the sex of the individual. As I said, they're indistinguishable. They're identical whether you're looking at a male or a female karyotype. But the 23rd set, or paired set of chromosomes, again, do distinguish between males and females, for humans at least. And so in this case, what we see is for these chromosomes, this 23rd set, we have one that is called an X and one that is called a Y. So this would be a male. And for a female, you would have 2 copies of an X chromosome, of the X chromosome. So again, because this set of chromosomes, this paired set of chromosomes, distinguishes between male and female, we call those the sex chromosomes. So here's another representation of a human genome, represented in this karyotype format. So there's different ways, you can prepare these chromosomes for visualization under a microscope. You can use different stains and so on. So this is one staining method. Here's another. In this case, they used a fluorescent stain, but it's the same idea. Chromosomes 1 through 22. Again, are the autosomes. All those autosomes exist as pairs in a normal human genome, and are indistinguishable, whether you're looking at a male or a female. It's the 23rd set of chromosomes that are different. Here's XY for the male, and here's XX for the female. So the question is how do you organize these karyotypes? When you fix and stain these chromosomes for visualization under the microscope, regardless of the staining technique, you have to identify the paired sets of chromosomes so that you can organize this karyotype. So how do you distinguish between the chromosomes. So here's an example. The first characteristic that you would look for is size. You can see that these chromosomes vary in size. So you have some like human chromosome 1, which is very big. Human chromosome 22 is very small. So they're easy to distinguish. But then you can see, human chromosomes 1, 2, and 3, they all have a similar size. So how do you distinguish between them? So the second characteristic that you look for, I'm going to use 15 and 16 as examples. These are two, human chromosome 15 and 16 are similar size. However, they exhibit this constriction that is different here for chromosome 15 and here for chromosome 16. So even though the chromosomes are similar size, the placement of that constriction is different, and it allows you to distinguish between the two different chromosomes. Okay, so that constriction is what we call a centromere. And again, the centromere plays has an important function during cell division, specifically during mitosis and meiosis. And again, we'll talk more about that in an upcoming lecture. So if you look at that constriction, again, you see it's different between 15 and 16. It's also different if you're looking at, say, chromosome 9. So there's different placements for that constriction, and there's terminology that goes along with describing that difference. So if you have a linear human chromosome and that constriction, that centromere is in the center of the chromosome, such that you create two arms that are of equal length for that chromosome, you describe that as a metacentric chromosome or a metacentric placement of the centromere. If the centromere is slightly off center, so that now one arm is larger than the other arm within that single chromosome, you describe that as a submetacentric placement of the centromere. And if the centromere is really off center, so that it's almost but not quite at the end of the linear chromosome, here you have a really big arm now, and then a little tiny arm here. That's described as an acrocentric chromosome, or an acrocentric placement of that centromere. For each of these two situations, submetacentric and acrocentric chromosomes, where you have two arms of different lengths, this is a human convention now for describing human chromosomes. The larger of the two arms is referred to as the Q arm, and the smaller of the two arms is referred to as the P arm. Okay, so P is for petite, so it's the smaller of the two arms. So there is one other centromeric placement that is shown down here. You can actually have chromosomes where the centromere is actually at the very end of the linear chromosome, so you only have one arm. Okay, so this is what's called a telocentric chromosome, or a telocentric placement of that centromere. So we don't have, in our genomes, a human genome, we don't have telocentric chromosomes, but we do have metacentric. Human chromosome 1 is a good example of that. So the two arms are of equal length, so that's metacentric. All of these through here, 6 through 12, are a submetacentric placement, where you see, two arms of differing lengths. And then you have this group here, 13, 14, and 15, where the centromere is way off center. So you have a really tiny arm and then a really big arm. Okay, so these are submetacentric, 6 through 12. These are acrocentric, 13 through 15. So again, as I said, if you survey all of the other chromosomes in the human genome, we don't have any telocentric chromosomes, but other organisms do. Okay, so now if we wanted to, so for, say, chromosomes 6 and 7, so they're of the same size, they have the same centromere placement, so how would you be able to distinguish them? Well, this is where you use a third physical characteristic of the chromosomes to assemble these karyotypes. So let's go to another karyotype here. So we're talking about 6 and 7. Okay, so again, this is a different staining method than the previous two karyotypes that I've shown you. And you see through this particular staining procedure, you actually generate a banding pattern of dark and light bands for each individual chromosome. So that's a physical characteristic that allows you to distinguish between individual chromosomes. So you can see the two members of the chromosome 6 pair, same size, same centromere placement, and same banding pattern. And you look at 7, the two members of this, the paired set of chromosome 7, again, similar size, similar centromere placement, similar banding pattern. So you can easily distinguish based on this banding pattern, chromosome 6 from 7. Okay, so these are the 3 characteristics that are used to assemble a karyotype once the chromosomes have been visualized. You look at the size, centromere placement, and banding pattern, which then allows you to assemble the karyotype. So the next question is that within this human karyotype that we're using as the example, why do we have 2 copies of every chromosome? And most other eukaryotic organisms having the same type of genome organization, 2 copies of every chromosome, 2 copies of every gene, the diploid state. So why does that condition exist? Well, again, it has to do with the means by which we reproduce. So predominantly among eukaryotic organisms, sexual reproduction rules. So what happens through the a reproductive method involving 2 different parents, sexual reproduction, is that each parent will contribute half of the genetic constitution of their genetic constitution to their offspring. So therefore, within each of these paired sets of chromosomes, 1 copy is contributed through the maternal parent or the mother, and 1 copy is contributed through the paternal parent or the father. Okay, so here's a slide that summarizes generally this process of sexual reproduction, and I want to describe it 2 different ways. I want to describe it at a cellular level, and I want to describe it at a chromosomal level. Again, and the purpose here would be to introduce some terminology. So here's 2 individuals, 1 female and 1 male, multicellular adult human, the body consisting of trillions of cells. So there's a unique subset of cells. The cells that comprise the body are what we call somatic cells. Those are non-reproductive. However, within the gonads, either the testes or the ovaries of male and female respectively, there's a unique type of cell, a germline cell, that has the ability to undergo a unique type of cell division that we call meiosis. And meiosis produces reproductive cells, sperm on the male side and an egg on the female side. Okay, and again, this is occurring within specialized cells that exist within the gonads of the male and female parent. So these 2 reproductive cells will then fuse with each other through a process that we call fertilization and generate a zygote. Okay, that single-celled zygote then has the ability to replicate itself or duplicate itself through cell divisions, through many rounds of cell division, that is different from this meiotic process that I described earlier. So in this case, through mitosis, that single-celled zygote has the ability to create trillions of somatic or non-reproductive cells that then construct the body of the offspring. Okay? So that's describing sexual reproduction at a cellular level. What about at a chromosomal level? So what do we know about the somatic cells that comprise the body and these unique germline cells within the gonads that can proceed through meiosis? Well, all of those cells are diploid. So again, for the human, there would be 23 pairs of chromosomes, a total of 46 chromosomes in all of those somatic cells and within those germline cells, those specialized cells within the gonads. So again, within the gonads, those cells have the ability to proceed through a meiotic event to produce sperm and egg. Well, those sperm and egg are unique in terms of being able to reproduce the parents in that they are haploid in terms of their genetic constitution. So in other words, the process of meiosis has reduced the original diploid cells to a haploid state. A haploid sperm or a haploid egg will carry one copy of every chromosome that is unique to the human genome, so a total of 23 chromosomes. When those two cells fuse through fertilization event, a zygote is created, and that zygote is a restored diploid condition. So now, again, that single-celled zygote, through the fusion of the sperm and the egg, contains a diploid chromosome constitution. 46 chromosomes, one copy of every unique chromosome in the genome being contributed by the maternal parent, and the other copy being contributed by the paternal parent. And once that zygote is produced, again, that cell can undergo many rounds of mitotic cell division that maintains that diploid condition. So when you reconstruct the body of the offspring, all of the cells that are produced through mitosis reiterate that diploid state. Okay, so again, some of the terminology, I've already defined these for you previously. The idea of diploidy somatic cells, the bulk of the cells that comprise the body of a multicellular eukaryotic organism. There's 2 copies of every chromosome, 2 copies of every gene. Again, for the human example, the diploid number is 46. For corn, as a plant example, the diploid number is 20. Okay, so the haploid state, again, sometimes represented as a 1N condition. This is the state where the cell has 1 copy of every chromosome, 1 copy of every gene. So again, these are going to be cells that are produced through a meiotic event. And again, the haploid chromosome number for humans would be 23, as we've discussed. And for the plant example for corn, again, it would be 10. Okay, so going back, just to introduce some additional terminology here. So we'll just take this paired set of chromosomes in a diploid genome that represent the 2 copies of human chromosome 2. So again, as I've described them previously, physically, the physical characteristics of these 2 chromosomes are identical. So they're the same size, they have the same centromere placement, and they have the same banding pattern. So in addition to that, from a genetics perspective, they also carry the exact same type of genetic information, at least in terms of the types of genes. So in other words, from top to bottom, in each of these copies of human chromosome 2 within this diploid genome, there's a DNA molecule that runs from end to end, one long molecule of DNA that within it carries genes, individual genes. And for the 2 copies of chromosome 2, is that each copy carries the same types of genes and in the same linear order. Okay, so this is what we describe as being a homologous set or a homologous pair of chromosomes, because they're physically identical to each other and they also carry identical types of genes. Okay, so that would be different if you were to say compare human chromosome 2 to human chromosome 8. So obviously, their physical appearance is different from each other, size, centromere placement, banding pattern. But in addition, they carry completely different molecules of DNA within each of those chromosomes, 2 and 8. And so therefore, they're genetically distinct from each other. There's no similarity at all. Okay, so that would be comparison would be described as being comparing 2 non-homologous chromosomes to each other. So again, just for a more formal definition of this terminology, homologous chromosomes are pairs of chromosomes in a diploid genome or diploid cells that are the same in terms of their physical appearance and also in terms of their genetic constitution, at least at the gene level, the same types of genes and the same linear order of genes along the length of that chromosome. So non-homologous chromosomes are the opposite of that. So typically they are morphologically, the physical appearance is different and they are genetically distinct as well because the DNA molecules that they contain carry completely different sets of genes, completely different genetic information. Okay, so there's one other bit of terminology that I want to introduce here, and then we'll wrap up. So here's a homologous set of chromosomes. Again, remember, within a homologous set of chromosomes for a sexually reproducing organism, one copy is inherited from the father, and one copy is inherited from the mother. So those two chromosomes, again, are physically identical to each other for those three characteristics that I've described. They carry similar DNA molecules along their length, such that from top to bottom, they have the same types of genes in the same linear order. So if you were studying or investigating a specific gene, and you knew the localization of that gene on the chromosome, say this example here, this is what we call a gene locus. So the locus is simply the particular localization for a given gene on a given chromosome. And there are actually mapping strategies that we'll talk about in upcoming lectures that actually allow you to place genes in particular regions of particular chromosomes. So in other words, to determine a particular linear order for given genes within a given chromosome. Okay, so again, when you have a diploid organism, two copies of every chromosome, you have homologous sets of chromosomes. There are differences between them. So it's simply the idea that even though the two copies of the chromosome, the two copies of the genes carry the same type of gene, there can be different forms for that same gene. And this is what we call allelic variation. So just to use a Mendelian example, Mendel worked with the pea plant and he was studying the inheritance of flower color. And he had so all of the pea plants carry a flower color gene, but some of them carried a form of the gene such that you got purple flowers, and other strains of the pea plant carried a different form of the flower color gene so that you got white flowers. So different forms of the same gene that produce, different characteristics of the organism. Okay, so that's due to allelic variation. So here's just to give you two examples from the human genome, representative examples. So somewhere within the human genome on one of the 23 unique chromosomes is a hairline shape gene. So everybody carries two copies of the hairline shape gene, but there are different forms of that gene. So one form of the gene, one allele, conditions what's called a widow's peak, and the alternative form for that gene, a different allele, conditions a straight hairline. Okay, so it's the same gene, the hairline shape gene, but different forms of the gene will determine different physical manifestations of that genetic information. Here's one other example, tongue rolling. There's a gene in the human genome that determines whether or not you can roll your tongue. And again, there's two allelic variants for this particular gene, two different forms of the same gene. One form allows you or gives you the ability to roll your tongue, and then the alternative form of the same gene, the allelic variant for the same gene, prevents your ability to roll your tongue. So again, all of these concepts are going to be important for subsequent discussions that we're going to have when we describe the processes of mitosis and meiosis and some of the initial concepts of Mendelian genetics. So again, I encourage you to review all of this terminology that I've tried to describe here. So I'm going to wrap up this lecture at this point, and what we will get to in our next lecture is spending a little bit of time going into a fair amount of detail describing similarities and differences between the processes of mitosis and meiosis, the mechanisms by which dividing cells segregate replicated genetic information, and specifically nuclear localized genetic information within, eukaryotic cells or eukaryotic organisms. Okay, so you can look for that lecture to be opened up on Tuesday.